Horasmantuapurba di channel YouTube. Seperti biasa kita akan menantikan acara yang sangat menarik tentunya bagi kita semua. Terima kasih Tuhan, karena dalam pernyataan Tuhan Yesus kita dapat bareng-bareng mengikuti acara yang sangat menarik tentunya. Tapi sebelum kita melakukannya, kita harus mengikuti protokol kesehatan, memakai masker, menjaga jarak, dan mencuci tangan dengan air yang mengalir, pakai sabun tentunya. Dan tidak lupa menikmati makanan bergizi, agar asupan gizi itu dapat menunjang kesehatan kita. Sebelumnya saya ucapkan terima kasih teman-teman. Mari kita ikuti bareng-bareng.
It is the complement system. Complement system consists of a series of proteins which complement or augment the functions of the antibodies. Again, what are complement system? Complement system consists of complementary proteins. It's a whole multiple series of proteins. The multiple proteins. And what are the functions of these proteins? These proteins basically complement the immune system and complement the inflammatory reactions. Or we can say these proteins augment the immune system work as well as augment the inflammatory response. We will see in this lecture that what are the complement proteins, how they are activated, why they are activated, what are the advantages of activating these proteins, what are the benefits to human beings by activating these proteins, and what are the dysfunctions of these proteins. If these proteins are overactivated, what type of problems are produced to us, or if these proteins are underactivated, what happens to us. First we will talk about the normal function, and then we will talk about the dysfunction of the complement system. Let's start with the normal function. The first question is where the complement proteins are present and where they are produced in the body, right? Actually complement proteins are mainly produced by the liver cells. So the source of complement protein in our body is mainly hepatocytes. Liver cells, you know, they produce a lot of proteins. As everyone knows that liver cells synthesize albumin, liver cells, hepatocytes produce many coagulation factors. In the same way, liver cells also produce a series of proteins which are about 20 or more than 20 proteins, and those proteins are called complement protein. Let's suppose this is your circulatory system. Here is your circulatory system. Here is your liver, and liver cells are constantly synthesizing and releasing complement protein in circulation, right? These different complement proteins which are produced, right? Okay. These different complement proteins which are produced in circulatory system, right? There is not one protein, there are about 20 or plus proteins, and these proteins interact with each other. And do you think in a normal person blood who does not have any inflammatory reactions or any immunological reactions going on, do you think in normal person's blood complement proteins are present?
Yes.
What do you think?
Yes.
Yes, they are. Complement proteins are present in your blood all the time. Is that right? Basically, complement proteins are proenzymes. These are inactive proteins. These are inactive enzymes. They are constantly being produced by the liver, added to your blood, right? But they are activated only during the inflammatory reactions and immune-mediated reactions. Is that right? So, major source of complement protein is liver. That is what a good doctor knows. But there are some doctors who are great doctors. They know more than this. That some of the complement proteins are secreted by the macrophages. And another important exception. Complement protein number 1 is mainly produced by the GIT mucosa. That is an exception. Most of the complement proteins are produced by liver. But if you really want to know exceptional things, many of them are also produced by macrophages. And complement protein number 1 is mainly produced by GIT, present in your blood. Is that right? Now, let's see how they work. right? We take a practical example that there is a bacteria present in your tissue. Okay, we make one bacteria here. What do you think? It's coccus or bacillus? Okay, he has high general knowledge. He knows it's bacillus, not coccus, right? Cocci are actually circular, spherical, right? So this is one bacteria. And of course, let's suppose this bacteria has these antigens. These are bacterial. These are bacterial antigens. And you know, when immune system is activated, immune system make antibodies against the bacterial antigens. Is that right? Let's suppose you have made an antibody against this bacterial antigen, and you know, antibodies are basically, they are two heavy chains antibodies, and they are having two light chains. All these chains are held together by, yes, what are these? Disulfide bonds. Remember the structure of antibody. Antibody is a tetrapeptide. Antibody is a tetrapeptide having two heavy chains and two light chains held together by disulfide bonds. This is the basic structure of the antibody. Let's suppose when this bacteria entered in your body, it activated the humoral immune response, and eventually against these antigens, antibodies are produced, right? There are many types of antibodies, right? But antibodies which can specifically activate the complements, they are immunoglobulin belonging to M class, IgM, or immunoglobulins which are IgG. You know, there are 5 main classes of antibodies, right? There is IgM, there is IgG, there is IgA, there is IgE, there is IgD. The complement activating antibody classes are only IgG and IgM. Is that right? Let us suppose that against these bacterial antigens, our immune system has produced IgG or IgM. Any one of these is produced. Once your immune system has produced these antibodies, let's suppose here is your immune system. This is your immune headquarter, right? You must be knowing that macrophage present the antigen to T helper cells. Macrophage take up the antigen, present to T helper cell, and T helper cell convert the B cells into plasma cells. And these plasma cells secrete the, yes, what is it? Antibodies. Basically this antibody is tetrapeptide, but in a simple way I've shown it like this. Now these antibodies which are produced against this antigen, eventually these antibodies will reach here, and what will happen? These antibodies will, yes, they will react with the antigen. This is an antibody, and now this antibody is reacting with the target antigen. Is that right? When antibody reacts with the antigen, there is a change in this tail of the antibody. It undergoes a change. It is just like that thing that if a boy is standing, there is no change. But if we interact with something, there is a change in voice. In the same way, when antibodies are not with the antigen, they don't have a specific change. But as soon as this antibody molecule reacts with the antigen, there is a change in it. What is the change in it? Let me tell you. Let's suppose this is the Fc portion of the antibody, and Fc portion of the antibody has a special loop here, right? This point, these 2 points can activate the complement. But normally when antibody is not binding with the antigen, these loops are not available to the complement protein. But as soon as antibody bind with the antigen, these loops become exposed. We say antibody is activated. And here is the answer. You have a lot of antibodies moving in your blood, but they are not activating the complements all the time. Why they are not activating the complements? Because antibodies which are moving in your blood or antibodies which are present in your body fluids, they are without antigens. Until they are without antigens, they are not having the capability to activate the complement system. But as soon as antibody bind with the antigen, it undergoes a transformational change and it exposes special domains which can bind and activate the complement system. Advantage of this fact is that antibodies will not inappropriately activate the complement system, and antibodies will only activate the complement system when antibodies are sticking with the antigen. That means antibodies will activate the complement system only when there is a need of it. When there is a need of it. Now, as soon as these points are exposed, it means now this antibody, which is biting on the antigen, it is activating its tail. It is willing to do some function. Here, what is the function? To bind the complement protein and activate. Now, of course, if there are many bacteria here, there is some injury is going on. It means there is some inflammatory reaction here, and if there is some inflammatory reaction in this area, it means that endothelial cells here there is some inflammatory reaction going on, and during the inflammatory reaction, blood vessels are dilated, endothelial cells are shrunken, and there is increased permeability. Again, listen. Because in this area, bacterial invasion is there, and bacteria are damaging this area, so naturally there will be inflammatory reaction, and due to that inflammatory reaction, there will be vasodilatation as well as there will be increased permeability of microcirculation. So there will be more blood coming to this infected area, and there will be more proteins leaking out from vascular compartment into interstitial area of this tissue particularly. Because this becomes more permeable, so your sleeping dogs, who are your sleeping dogs? Complement proteins which are sleeping in the blood. Now they have a chance to come here so that they become active dogs. So what happens that from here, different complement proteins, they will come out, right? But yet they are not active. These are not active. They're sleeping. Now what happens? Actually, by chance, they will start hitting at this point. Whenever complement protein number 1, right, will hit over here, it will be activated. Is that right? Complement protein number 1 has 3 parts. Complement protein number 1 we can show like this, that this is complement protein number 1, right? And this has 3 areas, 3 legs. One leg is called Q, other is called R, third domain is called S. You know ABC, QRS, no need to be upset. And this QRS has nothing to do with the QRS complex of ECG. Is that right? So what happens, we say that complement 1 protein, right, it has 3 peptides or special domains, QRS. This Q point can interact with activated antibody. Antibody in complex with antigen. So what really happens, let's make it and see what really happens, that C1 will bind here, right? And when C1 will bind here, now you will tell me it has opened its eyes. It has become active protein or inactive? It has become active protein, right? And what are these points which are activating here? No, only Q is activating here. This is C1 which is activated. As soon as we come in touch with the antibody which is complexing with antibody, not every antibody, especially IgG or IgM. Now it becomes active. When it becomes active, this is C1 activated. Complement number 1. We put a bar. When we put a bar, it means it is active, right? So next time what happens? This is inactive complement protein, and this is active complement protein. So we can put a bar on it. So in the books, when you find there is a complement protein written with bar, it means it is which form? Active form. So this is also active form. When it becomes active, it calls some other friends. Come here, there is an antigen antibody activity going, you are needed here. So its friends will come. What will be the next protein coming here? Oh, not directly C3. It comes C4. right? Now, so second protein which comes over here, right, that binds here and become active. Is that right? Very happy. Because it is being activated by C1. So C1 activates the C4. It activates, this was C1. Okay, I will put it in black color. Or you want which color do we put here? Okay, this is C1. Now what is this? C4. Is that right? Why this protein is called C4? First of all, we should be thankful to the researchers that there are many complement protein, and we should be thankful and obliged to those researchers who did not put their names on these proteins. They just call it protein number 1, 2, 3, 4. And these numbers are assigned according to the sequence in which these proteins were discovered. So unfortunately, protein number 4 was discovered before the 2 and 3. So that is why it looks a bit out of order. Is that right? But this was the order in which they were discovered. Right. So complement 4 is activated by C1. Right. Once it is activated, normally, now I have to make another diagram here. Let's suppose this is an inactive complement protein. It's just like a baby. And normally it has a very special domain, and it is like a, what is it? It looks like, you have seen? It's apple, sure. So you can say this protein has two parts, a baby part and baby with apple. This complement protein has two parts, baby part, apple part. Or we can simply say baby for, B for baby, and A for apple. So it's B part and this is A part. So most of the complement proteins have A for apple, B for baby. Not difficult to remember. This A and B, when they are complex together, they are inactive. When they are stimulated, look, baby is sitting like this with an apple on its head, and it is activated, what will happen to apple? It will go away. So what we will say, that when it is activated, right, baby become active, right, but apple has gone away. What is left behind is, for example, complement, which fragment? Baby fragment, B fragment. And which fragment has gone away? Complement, A fragment. Is that right? So many of the complement, right, they consist of a complete protein, and when that protein is activated, some of the complement break down into two components. One component is called B component, other component is called A component. Is that right? Am I clear? B for baby, A for apple. But here B is not for baby. Here B is for the fragment which remain binding with the original complex. One fragment fall away, other remain bound. B for binding. Let's see what happens. This was C4. When C4 is activated, which component is lost? Yes, please. Right? So what will be this? C4 A. And what will be this? C4 B. So what we can say? That when complement number 1 is activated by the antibody, Fc portion of the antibody or tail of the antibody activate complement number 1. Complement 1 protein activates the complement number 4, and complement number 4 hydrolyzes or breaks down into two components. One remain bound with this. This is called fragment B. One fragment diffuses away into fluid form, and that is called fragment A. So apple goes away, baby stays there. Am I clear? Now, once this baby C4B is there, it will attract another baby. Now what is that baby?